Hello, in this video we will discuss about the atherosclerosis. So that means the plaque development within the artery or vein. So how it's possible? Here is the person which that's contain circulatory system. So in this way, for example, this is the basically atherosclerosis will be developed. How? There are many complications and there are many disease which can lead to cause. For example, here is our artery which that is contain a upper part is the endothelial cell, and tunica media is deposit the fat and other cholesterol, and there are a lot of things, and in this way the block formation due to the high blood pressure and the resistance will be caused, and the lipid addition will lead to rupture of this thrombus is known as the thrombosis. So on the other hand, here is you can see this is the carotid artery which that supplies the oxygenated blood toward the brain, and if the plaque will develop and rupture is known as thrombosis after the atherosclerosis, which that will lead to brain hemorrhage. So the brain hemorrhage is due to the rupturing of the plaque and move that clot into the another part of the veins and artery where the smaller narrow range. So in this way, here you can see that is the carotid artery, and this is the block the blood flow to heart attack. So that is why when block the blood flow, it means the block and heart will not pump that blood, and it means the ultimately death will occur. So anyhow, here is the enlargement of the heart is also due to the this complication and the enlargement and kidney failure, and there are a lot of things. Edema will be developed, and coronary artery disease can be happen. Why? Because there are many regions which that is used for the atherosclerosis. So how it's possible and how atherosclerosis will develop? So they basically the complication due to the abnormal sugar level, abnormal cholesterol, hypercholesteremia, hyperglycemia, and hyperlipidemia, which that will lead to develop the plaque. So let's begin to understand. Here is the artery. The first decade, first 10 years, it will not develop too much. So it required a long time. So in this way, here you can see the histological normal here is, and the macrophage infiltration and the isolate foam cell can be happen, but the next is the fatty streak formation. In this way the fatty streak will be developed, and the mainly intracellular and lipid accumulation into the cell. So remember about these things, it will be developed into the intracellularly and damage the cell, and the next is the intermediate lesion will be occur, and this intermediate lesion is the intracellular lipids accumulation. Small extracellular lipid pools also will be happen. So and the next is the fibroatheroma formation. In this way the single or multiple lipids core, fibrotic and calcific layers will be developed. And here you can see the intracellular lipid accumulation will be occur, and the core extracellular lipid formation, and the complicated lesion and the surface defect. On the other hand, the thrombosis formation will be occur due to the hematoma, hemorrhage formation after the rupturing of the plaque, and this plaque is covered by the plug is known as basically is the plug, or we can say that is basically after third decade to the fourth decade. So that development is the hard scar tissue or the collagen deposition and fibrin for tightness. So here is the artery structure. Inside is the tunica interna, and it's contain endothelial cell inside, and inside is the lumen, while on the other hand the smooth muscle cells are present in the tunica media, and endothelial cells are present in the tunica interna, while the tunica externa is basically the upper part, and this upper part is basically the cover, and this is tunica externa, which that is elastic muscle, oblique muscle, as well as other which that is sportive and used for the flexibility. So anyhow, here is the endothelial cell surface, and the bit down is the tunica, media. This tunica media is basically used for the smooth muscle cell differentiation into macrophage. So it can be converted into the macrophage, differentiation into macrophage, and it can be movable. While on the other hand, here is you can see this is the pancreas. So let's begin to understand step by step atherosclerosis process and mechanism. So this is the insulin produced from the pancreas on the response of higher blood sugar. But in the case of some diabetic person, the insulin will not produce. And in this way, the insulin will not produce, so the glucose will not absorb due to the insulin binding receptor will not work, and the GLUT channel will not open. So the glucose remain high in the blood circulatory system. But the endothelial cell of the blood vessel, which that is not use the insulin and directly diffuse into the endothelial cell. After the entering to produce more ATP and reactive oxygen species and reactive nitrogen species and produce the chemokines, which that is the chemotactic movement, the positive chemotactic movement will lead to cause inflammation. So this is the endothelial cell damage. So here is the low density lipoprotein are circulate in the blood circulatory system and will invaginate due to the weakness of the endothelial cell due to the reactive oxygen species produced, and that will be damage the endothelial cell will lead to entering the low density lipoprotein where the space will be occur, and in this way the reactive oxygen species will produce and low density lipoprotein will be oxidized. So after the oxidized low density lipoprotein, which that is contain lipid, will be endocytosed by the scavenger receptor of the macrophage 1. So after this, here is the CD36 molecule as well as the co-molecule is co-receptor molecule, and the scavenger receptor A will bind and endocytose this lipid, and further it's going on. And in this way the foamy cell formation. After the foamy cell formation, you should know about that. This foamy cell formation is the basically living, but it is the precursor of the apoptotic cell. So it's contain toll-like receptor 4, 6 as well as 2 and MYP88 and the TRIF and node-like receptor P3 and CD40 and CD40 ligand, which that will increase the necrotic factor kappa B gene to increase the necrotic factor kappa B. What is the function of necrotic kappa B? That is lead to death of foam cell, and so that is here you can see that the matrix metalloprotease is also stimulated by this, and the macrophage will be stimulated to foam cell. This will be proliferate, and there are a lot of macrophage which contain foam of the lipid, so the foamy formation will be occur. Anyhow, I hope you are getting this concept. So this foamy cell will produce cell-to-cell proximity, fast ligand and nitric oxide and tumor necrosis factor alpha, which that will lead to necrotic cell death of the foamy cell, which that produce the necrotic core and apoptosis of macrophage and smooth muscle cells colony. Because the smooth muscle here you can see, the smooth muscle will move out due to the growth factor. This growth factor used for the proliferation and migration of the smooth muscle. Due to the rupturing of the membrane, this internal elastic lamina which that will be ruptured, and after this rupturing the growth will be increased, as well as the smooth muscle cell also produce the trigger the KLF4, the Krupfer like factor 4. So here is you can see this is the necrotic core apoptosis of macrophage and smooth muscle cells due to the proliferation of the smooth muscle cells. So here is the interferon beta and toll-like receptor 2 and endoplasmic reticulum stress will be increased, will lead to cause more cell death due to the necrotic mean the accidentally injury of the cell. So the problem of the cell will lead to cause necrosis. So the apoptosis of the necrotic apoptosis. So here the plaque will be developed, and this plaque will be bound with the fibroblast, which that will be recruit, and this fibroblast will deposit the fibrin as well as other elastin, and there are many type of protein. While here the matrix metalloproteases will be activated through foamy cells, and this foamy cell will trigger the degradation and remodeling of extracellular matrix. After this, here is the basically you can see this is the selecting, and this is will be recruit more monocyte. So here is a protein selectin glycoprotein 1, which that will bind, and as selectin will bind, and a VLA4, which that is a very late antigen bind. And in this way, after binding with these factors and this molecule receptors, after this binding, it will be invade into the plaque site. And this plaque site is basically, here is the red color is showing that here this is a not correct side, my mistake. So you can suppose that is a red color. So the red color of the endothelial cell, which that is the activated and the damage, while this orange color is the fibrous cap formation, and this will be inactive, the basically endothelial cell. And here is the, on the other hand, the basically here is the monocyte, and this is p-selectin glycoprotein 1, which that will trigger the chemokine ligand 21 and 19, which that will trigger the CD4 cell, T cell, helper cell, and CD4 will produce tumor necrotic factor alpha and interferon gamma, which that will lead to increase more, stimulate the monocytes. So on the other hand, the vascular cell adhesion molecule 1 and the cellular adhesion molecule 1 will be stimulated by the collagen and fibrin and extracellular matrix, which that is yellowish color is the extracellular matrix containing the fat and lipids and cholesterol, which that will be developed, and the fibrin and collagen, which that will be developed, and the hardening of this plaque formation. And here the fibrous cap formation will be occur, which that is the bulging out. And in this way, remember that let's begin to understand vascular cell adhesion molecule 1 and cellular adhesion molecule 2, which that is used for the invasion of further monocyte. So the monocyte recruit adherence and differentiation into macrophage. After this, the infiltration, the foam cell formation, the pro-inflammatory environment will be occur. And in this way, the pro-inflammatory environment will lead to cause the inflammation, advanced necrotic core formation due to the unstable plaque. That is the atherosclerosis, and that is the unstable plaque. Instability means that different factors, necrotic factor, and there are many factors, and the inflammatory factors, which that will lead to cause the rupturing of the fibrous cap. After this rupturing, the out movement of the plaque will lead to inhibit the blood flow, will lead to cause heart attack. So that's all things about the heart attack.